Good morning. I'm listed as the facilitator, but I'll tell you who the facilitators really are. It's Ken Burns and Jane Brockman, Dan Purich, Jamie Ellis, and David Bloom, who found my students for me. So thank you, guys. Today we have Mitch Gulkis, who is going to speak to us about DNA and the characterizing nucleic acids and enzymology of human base. He's going to put it in easier terms. I've told him to please tell me about DNA for an engineer to understand. So, Mitch, thank you for coming. We're glad to have you.
Awesome. Thank you guys so much for the invitation. I appreciate the opportunity to speak a little bit about my research. My name is Mitch Gulkis. I'm a rising third-year PhD student in the lab of Dr. Malika Kaglian at the Department of Biochemistry and Molecular Biology at the University of Florida. And we study base excision repair. And before we get into that a little bit, it's a DNA repair pathway. And before we talk about DNA repair, let's talk about what does DNA do? So DNA is a class of biological macromolecule that is formed by a polymer of different nucleotides. And these nucleotides encode for information. Specifically, there's these things called base pairs, where there's a nucleotide up top and a nucleotide at the bottom. And together, they code for different information. Now, DNA is often referred to as the book of life. It's the instruction manual that cells use to encode everything that they do, and really everything that we are is encoded in DNA. In humans, we have 3.4 billion base pairs. So it's a pretty long book. The central dogma of molecular biology states that the instructions in DNA are converted first into a copy and then into the actual functional product of what the DNA is telling the cell to do. So first, DNA is transcribed into mRNA. I think we've all heard a little bit about RNA in the last couple of years, but RNA is the copy of the DNA and it tells the cells what to do. Y'all hear me? Okay, cool. And from the mRNA is then translated into proteins. And proteins are the actual functional unit. They're what does the actual action of what the DNA is encoding. However, unlike a textbook, DNA is susceptible to damage. It can be damaged by a lot of factors that I'll go into later. But damage in the DNA causes damage in the mRNA, which causes damage in the protein. And damaged proteins can have different functions than they're supposed to have. And when proteins go awry, that can lead to human disease. So how do we get these mutations? One of the prevalent ways we get it is during replication. So when a cell divides, it needs to replicate its entire genome. And during that process, errors can arise. As you can imagine, even if the enzyme that does that is very good, there's 3.4 billion processes. And even if it's 1 in a million, that's still a lot. And another very prevalent thing is DNA damage. DNA is a molecule, like any other, that is susceptible to chemical damage. So DNA damage is a naturally occurring phenomenon. And when I say damage, I mean a chemical modification to the DNA that changes its function. And to show you how prevalent it is, I'd like to direct your attention to the screen. So, So it's a pretty common process. Ballpark, we generally say it's between 50,000 to 100,000 sites of DNA damage per cell per day. And so, that, as you can imagine, that adds up rather quickly. So without a way to repair DNA, none of us would be sitting here. But first you might be asking, why does the DNA need to be repaired? Other biological macromolecules, such as the mRNA and proteins, aren't repaired. If they get damaged, they're simply degraded and new ones are made. However, since each cell only has one copy of its genome, that's not an option for DNA. It needs to either be repaired or the cell will die. So here are a couple classes of things that can damage your DNA. Some of these are endogenous, so that means within the cell, they're naturally occurring as part of the cellular's life cycle. So these can be either oxidation, alkylation, hydrolysis, as well adduct and mismatch formation. However, those are all naturally occurring, and cells are pretty good at dealing with those. But there's some exogenous factors that can be a little bit more damaging, such as ultraviolet light, which is why we all wear sunscreen outside, hopefully. And then there's ionizing radiation, chemical carcinogens, such as those found in fossil fuels and cigarette smoke. However, sometimes we can exploit DNA damage and will actually damage the DNA of cells on purpose. And we do this oftentimes in cancer treatment using chemotherapy and radiation therapy to overwhelm the cancer cells and kill them. So another thing is that DNA is damaged in many different ways, and they're all chemically distinct from each other. And since they're chemically distinct, they can be caused by different actions. And being chemically distinct, they require different repair pathways to fix. One repair pathway couldn't, for example, fix a DNA protein crosslink and a photoproduct. That would have to be different repair pathways. And unfortunately, errors in these in DNA metabolism can cause reduced genomic stability. And genomic stability is what we describe as how the information is stored and how stable it is being stored. And decreases in genomic stability lead to cell death, human diseases, mutations, and cancer. Luckily, being multicellular organisms, we've evolved several repair pathways capable of repairing these. There's 5 major DNA repair pathways, and there's a couple minor ones. But the 5 major ones are mismatch repair, which repairs replication errors caused by faulty replication. There's base excision repair, which repairs single strand breaks and single base damages. And then there's non-homologous end joining and homology-directed repair, which repair double strand breaks and interstrand crosslinks. And finally, there's nucleotide excision repair, which repairs interstrand crosslinks and bulky adducts. So each one of these in its own right is a field of study, so we don't necessarily have time to talk about all of them. So I'll be focusing on base excision repair, which is what my lab studies. Base excision repair is a really good DNA repair pathway to study because these 2 types of base damages, single strand breaks and single base damages, are some of the most frequently occurring damages to the cell, with single strand breaks happening around 10,000 times per cell per day. And in addition, it's a fairly simple DNA repair pathway, which really lets us tease apart everything that's happening. So to go into that, the base excision repair pathway is an enzymatic pathway where enzymes are proteins that have a chemical function. And in this, there's 4 enzymes. So in the first step, DNA gets damaged. And I'm going to represent that using this little red star here. It's first recognized by a DNA glycosylase, which is an enzyme that cleaves that bond and releases the damaged base. However, now there's still no base there, so we still don't have proper coding potential. That's then recognized by APE1, which cleaves the DNA backbone. Then DNA polymerase beta is a bifunctional enzyme, so it does 2 things. It first removes that overhang and then fills it using its polymerase activity. And finally, a DNA ligase ligates that, ligates the nick and completes repair. So our lab's mainly interested in the last 2 enzymes in this pathway, both DNA ligase and DNA polymerase beta. And for the remainder of my talk, I'm really going to be focusing in on DNA ligase. But first, I'd like to take a brief detour to DNA polymerase beta and what that does. So DNA polymerase beta is a eukaryotic polymerase. A polymerase is an enzyme that extends DNA. It's what copies the DNA and adds new nucleotides to a DNA chain. It's the smallest one that humans have. And partially because of that, it's an error-prone polymerase. It makes 1 error every 5,000 nucleotide incorporation events, which might not sound high, but the ones responsible for replicating the genome make 1 in millions. So it's about 200-fold less accurate than the replicative polymerases. And that's mainly because it lacks a proofreading domain. Whereas the replicated polymerases have a specialized domain that if they make an error, they can go back and remove it, like a backspace would on a computer. This enzyme, unfortunately, doesn't have that. So it's able to incorporate faulty nucleotides, which I'll get into a little bit more later. First, now to introduce the star of the show, the DNA ligases. This is a class of enzyme that we're really interested in, and I'll get into the mechanism in the next slide. But ligases are involved in many pathways, not just base excision repair, but also all the other repair pathways that I mentioned earlier, as well as in DNA replication. In humans, we have 3 DNA ligases, 1, 3, and 4. Maybe asking where did 2 go? It turns these were named in the order of discovery, and scientists thought they found ligase 2, and turns out they were wrong. But to be historically accurate, they named the next one 3. They all do the same chemical reaction. They all do Nick sealing, but they have slightly different biological functions. So in humans, DNA ligases are all ATP-dependent enzymes. ATP is a chemical, which is often referred to as kind of like the energy of the cell. It allows enzymes to do reactions that are not thermodynamically favorable. So ligases all follow a 3-step conserved mechanism. In that first mechanism, ligase will bind ATP and attack the alpha phosphate. And this forms a ligase 1 AMP intermediate, just shown here. This is what the AMP looks like, and it's bound to an active site residue of the ligase. In the second step of the reaction, the ligase is going to transfer that AMP to the DNA and form this covalent DNA intermediate. And in the final step, ligase is going to catalyze the attack of this atom here onto this atom. And that's the actual chemical reaction that ligase is doing, is it's forming this new phosphodiester bond. And that results in AMP release, and the nick is sealed. All ligases follow this conserved pathway, but bacterial ligases use a different cofactor. Same process, though. It's also important to note that ligases can fail. Ligases are very sensitive to the DNA base pair at this 3' end, and if something gets damaged or mismatched or not appropriately incorporated there, the ligase can transfer the AMP to the 5' end of the nick, but fail to ligate. If that happens, it results in a persistent strand break because ligase has to first get rid of this AMP before trying again. It can't just pick up where it left off. Another enzyme has to come in and clean this DNA end. So when you get this ligation failure, you get these intermediates that are crucial to genome stability because they result in accumulation of single strand breaks. And single strand breaks are generally pretty toxic. So another thing that really emphasizes what's important about DNA ligases is, well, what happens when they go wrong? In mouse models, we have the ability to knock out genes. So when you remove any of these 3 genes from mice, they all die in utero. It's embryonic lethal. So if any of these enzymes are missing, it's incompatible with life. However, it is possible that you have these enzymes, but they're dysfunctional or something's wrong with them. In that case, you get, unfortunately, diseases. One of the ones that we study is ligase 1 deficiency disease. And this is characterized by abnormalities in hematopoiesis, so the generation of blood, lymphopoiesis, which is the generation of your immune system, and severe immunodeficiency. So unfortunately, patients with ligase 1 disease deficiency don't make it very long. So to zoom in even farther, for my thesis work so far, I've been focusing on these 2 active site residues. So proteins are polymers of amino acids, and specifically we call them residues because they've been linked in a chain. But specifically, I'm going to be looking at these 2 residues. There's F635, where F is the code for what amino acid it is. It's phenylalanine. And 635 represents what position in this chain it is. It's the 635th amino acid. I'm also going to be looking at F872. And a previous paper back in 2004 suggested that these residues might be important for the ligation reaction. And so we're going to look and see if they actually are. You'll note that the bond that ligase makes is between 3' and 5' end. And in space, these two residues are the closest residues to the 3' and 5' end. So we believe that these residues are important for ligase's ability to actually do its function. Another way that we use to kind of see whether we think a residue might be important is we can align the sequence of our protein with other proteins. So it's a little hard to see here, but these are different colors, I promise. But we align the sequence of our protein, DNA ligase 1 from Homo sapiens, so from human, with ligase 3 alpha and 4, and we see that this residue is the same. In addition, we see that it has the same residue in mice and yeast, as well as a eukaryotic virus. However, in bacteria, it's a different residue. So we generally refer to this as being conserved back to yeast. We also did this for 872, and we found that this residue is conserved even farther all the way back to bacteria. So as we evolve, there's selective pressure to evolve better enzymes. But this residue didn't change, which likely means that there's a selective pressure to not change it. And that likely means that it's important for function. So that's one of the things that bioinformaticians use to kind of predict what's important to study. Because our protein's 919 amino acids long, you can't study them all. That would take too long. So first, what I wanted to do was biochemically characterize these proteins. Specifically, one way we do this is to see a function of an amino acid is what happens if we remove it. If it's doing something important and we remove it, we should be able to pretty easily see something go wrong. But in order to do that, first we have to purify these proteins. So what we can do is we have a control over bacteria, so we can make these bacteria express specifically our protein of interest. We can then use this instrument here, which is a liquid chromatography system, to separate our protein of interest from all the other proteins in the bacteria. Specifically, we're going to use 3 different columns to separate them, and each one of these columns separates proteins based on a different aspect. So this is somewhat similar to if you had a box of Legos and you first picked out all the red ones, then you picked out all the square ones, then you picked out all the 2x2 square ones, and then you're finally left with just 2x2 square red Legos. So that's similar to what we do here. Once we have these purified proteins, we can test their function. And in order to test their function, we're going to do a ligation assay. Ligases ligate, so it makes sense to test if they can. In order to do that, we'll need 3 things. We'll need a nicked DNA substrate. So this is a piece of synthetic DNA that's commercially available. There's companies that synthesize these. That's what the DNA is what the ligase will act on. We'll need our actual ligase that we're interested in, and we'll need a reaction buffer because ligase needs several other things to do its function that I'm gonna gloss over real quick. We'll then incubate those in a test tube at 37°C, which is body temperature, and we'll stop the reaction at different time points to monitor its progression. We stop it using a dye that denatures the protein and renders it inactive. We then run those samples on DNA sequencing gels, which are literal gels, and these gels separate DNA based on their size. So smaller fragments of DNA will run faster than larger fragments. And by doing that, we can then image them on this instrument here, which is a fluorescent laser scanner, and that allows us to visualize the DNA. We then have software available that can quantify the fluorescent signal, and that allows us to analyze its function. So a little bit more into that. Here's a little schematic of what we're doing. So we have our nicked DNA substrate, which is formed from 3 synthetic oligonucleotides. We have an 18-mer, which is 18 nucleotides long, a 16-mer, and a reverse complementary 34-mer. It's important to note that this 16-mer is labeled with FAM. FAM is a fluorophore, which means when you blast it with light, it glows. So we can see it. We then incubate it with ligase 1, all of our different mutants that we're going to be testing in this study for varying amounts of time. And that produces two products. It either produces a 16 mer plus AMP, and that's that intermediate I was telling you about earlier, where the ligase has transferred the AMP to the end of the DNA, but hasn't completed it yet. So this is an intermediate in the reaction. And then there's the full reaction product, which is what happens when the reaction is over. And these are kind of what these gels look like. To walk you through it, the smallest DNA migrates the farthest. So as is the farthest down the gel, this is the substrate, it's a 16 mer. This band here is the 16 mer plus AMP, and this is the 34 mer. And up here, you can see that we did 3 different substrates because we're going to be testing the impact of these mutations on different things here at this base pair. This is the base pair that I was mentioning earlier that is sensitive to mismatches or damages. So we're going to see how ligase can normally handle damages or mismatches, and then how it can handle it after we've mutated it. So these gels are great and all, but they're a little hard to look at. So we quantify them and make bar graphs, which are a little nicer to look at. And I should mention that all 3 of these are mismatches. So these are all errors that polymerase beta might incorporate during base excision repair. And you can see that ligase is able to ligate them with different efficiencies. For example, your TC mismatch, ligase can ligate that one fairly well, going almost to completion in 10 minutes. But the CC mismatch can only go to maybe 30% of the reaction. So when we do this for all possible base pairs at the 3' end, you'll see that there's a varying efficiency for each one. This diagonal here is the canonical nucleotides. So this is what's supposed to be there. This is what is there if polymerase beta did its job right. And you'll see that ligase can ligate all of those, which is great. It's supposed to. However, you'll note that there's specifically 3 other mismatches that seem to evade discrimination by ligase and can be ligated as efficiently as a canonical nucleotide. There's GT, CT, and TC. So that's a little bit interesting that there's these nucleotides that escape discrimination. But we wanted to see what happens to this profile when we mutate F635 and F872. So it's a pretty clear change. Where before we had ligation product and we got an accumulation all the way up, all the way up to 80–90%. Now all of our substrates are down to 5%. So whereas this was all the way up to pretty much completion, now it's only down to 5%, because I'll also point out this axis changed. And when we did this for all 16 substrates, we were wondering, well, did we just make a ligase that's not capable of ligating anything? Or is there something else a little bit more going on here? And we found there's something a little bit more going on here, because with this mutation, ligase 1 is still able to ligate all the canonical nucleotides, which are those are the substrates that it's supposed to ligate, but now it can't ligate any of the mismatches. It can't ligate any of the things that it's not supposed to. So we're really interested by that, and we decided to look at it a little bit farther, and we made a second mutation at this 635 site. We made phenylalanine to leucine mutation, which is a more conservative mutation. Leucine is more closely related to phenylalanine than alanine is related to phenylalanine. And we were wondering if this mutation you might see an intermediate phenotype. However, it seems like it's not the case, as this mutant is also capable of ligating canonical nucleotides, but not mismatched. We also wanted to go ahead and characterize 872. So we did the same thing for our other 2 mutants. And in this case, we saw the same phenotype where we saw ligation of canonical, but no ligation of mismatches. However, you'll note that some of these canonicals are starting to approach a lower efficiency, which might suggest that this residue is more important than 635. And this is really interesting, but this is about as far as we can go using biochemical methods, because biochemistry is the chemistry of biological systems. We've investigated what these mutations do on the chemical process that ligase catalyzes, But now we'd like to get a little bit deeper into the nuts and bolts of what's going on. And to do that, we're going to use a biophysical technique. Specifically, we're going to use X-ray crystallography. This is a biophysical technique in which you purify your protein and crystallize it. And I mean that literally, they're crystals, like salt crystals. You subject those crystals to a high-intensity X-ray beam, and that causes an interference pattern between the crystal lattice and the X-ray beam, and you get a diffraction pattern. Using some math, you can turn that diffraction pattern into an electron density map. An electron density map is a 3D representation of where the electrons are in your structure. And by knowing where your electrons are, you can then model in where your atoms are. If you can model in where your atoms are, you can get a final 3-dimensional structure of your protein. This is really important to do because protein structure informs their function. These aren't simple catalysts. They're very complicated, and how they're structured impacts how they function. Now, it's no easy feat to crystallize proteins. I was lucky. The enzyme I'm working with, we already knew how to crystallize it. But when you're trying to start a new crystal or a new protein and crystallize it, it's not uncommon for crystallographers to screen thousands or tens of thousands of conditions because you have to coax your protein in this droplet here at just the right concentration that it's soluble, so it's still in solution. But then you have to slowly remove the water at just a controlled speed that your protein crystallizes. If it's too fast, your protein will come out of solution, but it'll be a gunk, and the gunk's not useful. If it's too slow, it might take 20 years to crystallize, and hopefully I'll be graduated by then, so it's a little too slow. But it's a very difficult process. This is actually the bottleneck of crystallography in modern times. The diffraction used to be and the computation used to be the bottleneck, but computers have gotten pretty good, so it's not the bottleneck anymore. You also need a very high concentration and a very pure protein. So you see here, I have 30 mg per mil, which is high. You can just take my word for that, that's pretty high. And then there's only one band on this gel. This gel shows all the proteins in my sample, and you can see there's only one. So here's what an actual crystal looks like once you grow them. They're quite small. You'll note that the scale bar down here is 100 micrometers. For reference, human hair is about 50 micrometers. So this is about 3 human hairs in length. They're fairly small. And the hardest part for me personally was learning how to loop these crystals, because we have to manually go into the drop and use these little lassos to lasso the crystal. And they're brittle, they'll break. I've broken hundreds of them. It's quite frustrating, but once you get the hang of it, you can get some pretty nice pictures like this. This crosshair here represents the X-ray beam that we're going to shoot our crystal with, and that's going to create these diffraction patterns. Each one of these spots is constructive interference from the crystal lattice, and each one of these spots tells us something about the 3-dimensional structure. I'll show you the formula, but won't go into it. I'm not going to make you guys do math at 10:30 in the morning. But using these spots, we can go from reciprocal space, which is what this diffraction pattern is in, to real space. And luckily nowadays, the computer does this. But one of my professors who taught me said that in the old days, he did it all by hand, and it took him 2 years. And now it takes me 20 minutes. So, love computers. Another thing that we're going to need is a X-ray beam. And this is going to need to be much higher intensity than the kind that you get at the doctors. Much, much higher. So much so that we're going to need a particle accelerator. And the one we use is the advanced photon source at Argonne National Lab. Here I've just screenshotted Argonne National Lab, and you may be wondering where it is. If you're ever looking at maps and wondering where a particle accelerator is, it's the big circle. Zooming into that, just for scale, I think it's still kind of hard to comprehend how big this instrument is. So zooming into this corner here, you can see those are cars. This ring is 1,225 feet in diameter, which is about 4 football fields. And using this technology, we accelerate electrons to 99.999999% the speed of light, which is kind of hard to think about. That's equivalent to these electrons are moving at a speed which would circumnavigate the Earth 7.5 times every second. So they're going pretty fast. And when these electrons curve, they lose energy. And by conservation of energy, they have to give that energy off in a form. And how they give it off is in X-rays. And you'll see each of these little these things here, these are all places where they've channeled the X-ray beam into a focused spot. So this synchrotron has 68 beam lines operating continuously. So that's really a remarkable piece of technology. We don't just use it for protein structures, you also use it for materials research. Researchers make better solar panels here, better drugs, better materials, better things to make your cars out of. It's a really remarkable piece of technology. Where is that? It's about 30 minutes outside of Chicago. And the US, it's funded by the Department of Energy, and the DOE has 5 of these in the US. But this is the best one, in my opinion. It's also the only one I've used, so I might be biased. But once we've done all this, and we're able to get a 3-dimensional structure of our protein. So these are my actual proteins. This is the 635 mutant, and will be in this row. And the 872 mutant is in this row here. And I have them in complexes with different types of DNA. So this DNA, AAT, that's one of those canonical ones. If you remember back to the dot plot, these are the ones that polymerase beta is supposed to insert, and they're all blue. And you'll see that for both mutants, the AMP is attached to the 5' ends of the DNA, which is step 2. This is in line with what we've previously known and previous structures of this enzyme. We also complexed our 2 proteins with the AC mismatch. This is a mismatch that was not ligated well by our 2 proteins. And we can see that in these structures, the AMP is still attached to this active site protein residue, which is step 1. So this kind of explains why these enzymes are not capable of ligating these 2 substrates. I'll go into that a little bit more in the next slide. And finally, we solve the structure with GT. The GT mismatch is a really interesting one because it's able to be ligated by the wild type enzyme, so the enzyme with no mutations, but it's not able to be ligated by either of our mutants, which means that with this, by testing this mismatch, we're really trying to get into what's going wrong, because normally it can do this, but now it's not. And one of the ways that we really get a lot of insight from these structures is not just by looking at them alone, but it's by looking at them together. So in this figure here, I went ahead and superimposed 3 different protein structures. So the one in gray here is the wild type enzyme. The green is the enzyme with the 635 mutation, and the orange is the one with the 872 mutation. All 3 of these proteins are able of ligating this substrate, and they were all captured in step 2 of the ligation reaction. And it might be a little bit hard to see, but all of these superimpose quite well. You'll note that the 3' end overlays with each other very closely. The 5' end overlays very closely, as is the AMP. This is in stark contrast to the AC mismatch, where these changes might look subtle, but subtle changes can do big things to enzymes. So here in magenta, I have the wild type enzyme, then in dark blue, my 635 mutant, and in light blue, the 872 mutant. And you can see that these structures do not overlay well. You'll note, whereas if you look at the magenta structure, compared to the other 2 structures, they're not overlaid nearly as well. And we hypothesize that this is caused by mutation of our 2 sites, which causes DNA end instability. So the ligation reaction is very sensitive to the geometric constraints of nucleophilic attack. And these two residues, we believe, are really important for aligning the two DNA ends so they're in the right position to form a bond. If they're not in the right position, doesn't matter how hard you try, they're not going to form a bond. They need to be close enough and at the right geometry. We also saw this for our GT, and this is the one we're really interested by, because it can normally be ligated, but with our mutant, it cannot be. And we kind of see the reason for that here. Whereas we have in blue, we have this catalytically competent arrangement. I'm not showing it here, but if we overlay this blue structure with these, they overlay quite well. But if you look at the blue compared to this pink here, you'll see that we have a pretty gross rearrangement of the active site. And that's what's blocking catalysis. So by doing these biophysical experiments, we're able to explain what we observed in our biochemical experiments. Combining them, we're able to see the impact of what these residues are on ligation. By mutating these residues, we create DNA instability, which prevents ligation by causing suboptimal catalytic alignment. And of course, there's more to be done here. There's other experiments we could do to further prove this. But that's how far I've gotten so far. And you might be thinking, cool, so what? Over the course of this talk, I've zoomed in several times into something that's pretty minute. I'm studying 2 amino acids in one protein out of 919 amino acids in the protein out of the tens of thousands of proteins that the human genome encodes for. So what? But I'd like to argue that the contribution of these two residues to DNA ligation help us understand the DNA ligation mechanism better. By understanding the ligation mechanism better, we understand base excision repair better. By understanding base excision repair better, we have a little bit more of an understanding of DNA repair. And by understanding DNA repair better, we understand human health better. So, oftentimes you'll read about huge breakthroughs in the field of science, and those are great. I wish to do that one day, that'd be awesome. But a lot of times science is slow, methodical, thorough progress. And I hope to have convinced you that I've done a little bit of that. So with that, I'd like to acknowledge everyone in my lab. I'd like to thank my PI, Dr. Malika Kaglian, for all her support. Postdoc in my lab, Dr. Chun Tae, who really trained me on all the crystallography, as well as our collaborator, Dr. Vanderkoy, also in the department, for his help accessing the synchrotron light source. Finally, I'd like to thank you guys for your time and offer time for any questions.
Does anyone have a question? Here's an engineer who thinks he may understand part of it. Thank you very much. It's very
Certainly.
complex. Questions?
This is peripheral to your talk, but I have a comment. The other day on the news, I saw that with AI, they are able to do protein structure, 5,000 proteins, each one of which is a PhD student. But with AI, they can do it like that. Is that correct? I mean, would that help you in your work?
Yes, that is correct. In the last couple of years, machine learning has gotten really good. And so now there's a couple of groups, but one of the ones leading, there's AlphaFold, which is a company owned by Google. And there's another one it's David Baker's group at the University of Washington. And they're able to do some pretty amazing things. Yes, they are able to use computers to design new structures. However, it's still not perfect. They can predict new structures, but sometimes they get it wrong. One of the papers I read, they predicted the structures of like 3,874, I think, different proteins. And it turned out 3 of them were right. So
That's not acceptable.
But that's much better because you're able to screen all those and see which ones. And what they did in that paper is they created a protein that doesn't exist in nature. So while that may sound like a low hit rate, that's pretty phenomenal still.
So with that procedure, you would eliminate or bypass all that crystallography steps that you described and purification and all that?
I think the field's moving in that direction.
Yeah.
Currently, they're still considered structural predictions, and they have to be validated by
Right.
either crystallography or another structural method. But I wouldn't be surprised in 5-10 years if
Yeah.
this is a little obsolete.
Yeah. Well, thank you. You're doing great work.
Certainly.
I'm impressed. Thanks.
Thank you. I appreciate it.
An alternative approach, of course, to crystallography is cryo-EM,
Yes.
and you might want to just contrast the advantages of the two techniques, or how useful they are.
Certainly. So here we use X-ray crystallography, which has really been the workhorse, of structural biology for the last, we're coming up on 100 years. The first protein structure was 1958. So last 70 years. But there's a newcomer in the field. And as a scientist, we say new. It's been around for 20, 30 years, but that's still relatively new. And it's a cryo-electron microscopy. In this technique, instead of growing crystals, we're going to use an electron microscope to image individual molecules, individual particles. And we're going to do that for a lot of particles. I say a lot, I mean hundreds of thousands to millions of particles. We're then going to use computers to align all those images. And by taking a lot of pictures of something 2D, if you put all the 2D images together, you get a 3D image. That's a simplification, but that's the general gist of it. And cryo-EM is a fantastic technique. It allows us to bypass the crystallization step, which is what I told you earlier is really the bottleneck. However, there's some limitations to cryo-EM. Your protein complex has to be large enough. Currently, they're able to do proteins of about, depends who you ask, but ballpark 100 kilodaltons. But there's many proteins that are smaller than that are too small for the electron microscope to actually see. And in that case, you might need to do crystallography. In our case, our protein is 73 kilodaltons, but plus the DNA, it is about at the range that we could do cryo-EM. But we're able to get good crystals already. And crystallography is a little more tried and true, and practically speaking, it's cheaper. So we use that method. In the future, though, we do hope to use cryo-EM for determining protein complex structures, but that's something that we haven't gotten around to yet. It's a really fantastic technique, though.
So what were the 17 something or other sizes? What is that term?
So kilodalton.
Kilo what?
Dalton.
Dalton. Oh, thank you.
It's named after. Yeah, it's the atomic mass unit. Whereas one carbon atom is 12 daltons. So our protein is 73,000 daltons.
Thank you.
Mm-hmm.
Any more questions?
I think one point as a virologist that interests me and why I asked you about the cryo is that you talked about how small the limitation is, but actually you're going in the other direction, and crystallography has difficulty beyond a certain size, and that's one of the real potentials for cryo-EM is that you can take large molecular structures like a viral particle and actually get very good resolution in terms of determining the structure.
Yeah, certainly. Currently, the reason that the cryo-EM has that threshold is it's not any limitation in theory. It's a hardware limitation. The detectors just aren't good enough yet. But I wouldn't be surprised in the next 5 or 10 years if they get good enough and crystallography kind of becomes obsolete. That being said, cryo-EM does currently have some other issues with resolution of the structures. But that's, again, a hardware limitation, not a fundamental limitation. There's also cryo-EM is also computationally more intense, whereas I run all the calculations for my crystallography just on my laptop. One of the other labs in our department does cryo-EM, and they have to use a UF supercomputer. So it's a little bit harder, but again, computers are getting good pretty fast, so I wouldn't be surprised if it surpasses.
You mentioned the advantages of synchrotron radiation. Has anyone used neutron scattering or neutron diffraction, since it's more sensitive to the lighter elements?
Yes, they have. X-ray diffraction is the most common, but neutron diffraction is used as well. As you mentioned, the lighter elements diffract neutrons better. So in our structures, unless you get really high resolution, you can really only see carbon, nitrogen, and oxygen atoms, sulfur if you have them. But with neutron diffractions, you can specifically see hydrogens really well, because hydrogen atoms scatter neutrons very well. There are some downsides to neutron crystallography, namely that you need a larger crystal for it to work. And that's just due to neutron flux. Because if you have too many neutrons around, bad things happen. Bad things being like an atomic explosion. So bad things happen if there's too many neutrons around. So they use a lower and controlled flux, but you need a larger crystal for larger amounts of exposure time. Here, my crystal is about 100 microns long. You might need one 10 times that size for neutron crystallography. And I only exposed my crystal for 0.2 seconds each frame. So total, it's 72 seconds. From what I've heard, neutron crystallography can take days to collect a data set. But you get very good data out of it if you can do it.
Any other questions? Well, thank you very much, and I appreciate you coming and spending time with us. Next week, we are having a study of the African social velvet spider in Namibia. So
All right.
we're going to
Cool.
go from little tiny things to much bigger things. And I don't know how many people love spiders, but we'll see who shows up. Thanks again.
All right, thank you guys.